So we were able to remove over 400 genes and maintain a doubling time of just under 3 hours. So to put this into perspective, if you remember, I showed you that growth rate of Mycoplasma genitalium, which has a 15-hour doubling time and a genome size of 580 kb. So this is 50 kb smaller than that genome that's found in nature, and it grows about 5 times faster than that. So as you would expect, the minimal cell retains all the essential genes known for the core cellular functions. We partition these into 4 major groups where we have expression of genome information, preservation of genome information, cell membrane structure and function, and cytosolic metabolism. And what was interesting is 17% of those the genes, or about 79 of them, we could not assign them to any functional category. So if you look here, this is a blast map of all the proteins in our minimal cell and homologs found in other organisms. And we group these on the left where we're absolutely certain what the function of the gene is, to all the way to the right where we absolutely have no clue what it is. And so even though we don't have any clue what the function of that gene is, we know that it's essential for life. You delete it and you can't get viable cells. And what's even more interesting is that in many cases, some of these genes of unknown function are found throughout life, even in higher eukaryotes. So we see this as the next exciting phase of this project. We started with 149 genes of unknown function, which is about 1/3 of the genome. I think we have it down to less than 100. Part of that was through help from the scientific community, reading our paper and saying, hey, I have an idea, I think it might be this. And so I think we're down to just shy of 100 at the moment. Another one of our long-standing goals is to group all the genes on the minimal genome by biological function. So we refer to this as either modularization, or, to use a computer analogy, to defragment the genome. So you can imagine grouping by biological process. Some of these processes are just examples indicated here. As a proof of concept, we were able to take a 1/8 section of the minimal genome, modularize all the genes on that section, and show that the cells are still viable. And from what we can tell so far, it doesn't seem to be compromising growth rate in any way. So of course it's going to be more challenging, and we're going to learn a lot as we move on to the next stages, because a 1/8 section versus the complete genome is a big difference. So again, understanding the genome design principles. So we published this work in Science last March. So this is an effort that has been started and led by Craig Venter, Ham Smith, and Clyde Hutchison. They've been working on this for well over 20 years, and I've been fortunate enough to work with them on this project for the past 13 years. So in summary, for this part of the talk, so the creation of the SYN 3.0 minimal cell resulted in robust genome construction workflows and a chassis for scientific discovery. It's our genome synthesis and activation technologies that are now enabling the discovery of new genome design principles for minimization, defragmentation, and also looking at novel gene functions. So we always imagined, once we got to this point, that we would use the minimal cell as a chassis for learning how to build more complex and useful organisms for industry But as we looked at this, that cell is going to need, again, to use a computer analogy, a major software upgrade in order to get to that point. But I have no doubt that, as we understand the principles of how to design life from the bottom up, we'll eventually get to that point. We're not there yet. And there's still a way to benefit from the technology right now. It's more cost effective to go out in nature, find a more evolved starting point, and start applying our synthetic cell and minimal cell technologies to that more evolved starting point. And it was about 3 or 4 years ago that we found what that ideal starting point would look like. It's a gram-negative bacterium. It's called Vibrio natrigens. We call it Vmax. This is why it's an ideal starting point for us. So it's not a pathogen. You have to be able to genetically modify it if you want to do anything with it. It has a fast growth rate. In fact, this is the fastest growing organism in the world that's known. It has a doubling time of only 9 minutes. E. coli, for example, has a doubling time of anywhere between 20 and 30 minutes. It grows to very high density in low-cost medium. It grows in defined medium, so that allows you to better define the complete genetic system. So this is very much unlike Mycoplasma mycoides, where it has a lot of stuff in the medium, but we don't quite understand what all the components are. It has the potential for recombinant protein secretion. This is something that if you're going to make proteins in this organism and you can secrete them, it's a way to really facilitate the downstream purification processes. It has a natural transformation machinery, which allows you it really accelerates the strain engineering efforts that we want to do on this organism. And it's already compatible with the E. coli protocols, vectors, and reagents that are pretty much already entrenched in every single molecular biology laboratory. So a lot to like about this organism. So over the past several years, we've been developing a complete molecular biology tool set for molecular cloning and protein expression applications, and we published this paper last year if you're interested in learning more about it. So this work was brought to us by this guy here, Matt Weinstock, who works on my team at Synthetic Genomics. So shown here is it's a growth curve comparing Vmax in blue to several E. coli strains, including this one in gold, which is thought to be the fastest growing E. coli strain. After about 6 hours on agar plates in the lab, you can see Vmax colonies are clearly visible, but E. coli colonies are not. We're always thinking about, in a synthetic biology lab like mine, how can we speed up our workflows, really getting at how do you accelerate this design, build, test cycle. And just to walk you through the cloning process, which is something that that happens in just about every molecular biology laboratory every day. You have your DNA fragments, they get assembled. You then put them into a host organism, and you have to wait for colonies to form on plates. Usually you do that overnight. The next day you pick those colonies, and then another day you have to wait. So it's this waiting process then for the cells to grow up to a high enough density where you can then purify those cells and to purify the plasmid DNA out of the cell. So in E. coli, this is usually about a 3-day process, but with Vmax, you can do this now all in a single day from beginning to end. So huge time savings there. So for all of these reasons that I told you about Vmax, we're using it to develop our bacterial genome design software. where we want to make a Vmax genome that's completely minimized, it's defragmented, and it's recoded. So for industrial applications, some of the benefits are, so for minimization, you remove the genes and pathways that are not essential, and maybe they interfere with the desired process. For defragmentation of the genome, so as it's currently done, let's say you have to make 10 modifications to that genome to different parts of a pathway that you want to engineer. You make that change, and then you have to wait. You make the other change, you have to wait. Maybe with CRISPR-Cas9, you can now begin to multiplex and do this a little bit faster. But imagine if you had a genome where all the biological processes are already modularized, and then just all at once you could upgrade that biological function, replace that module with a new module, or maybe you have a combinatorial library of new pathways that you want to test and then select for the pathway that works the best. And then for recoding, this is a way to create a genetic firewall against bacteriophage infection and horizontal gene transfer. So we're building Vmax as a next generation chassis for biotechnology. E. coli is used to produce about a third of all the biologics that are made in the world, and it's still early days with Vmax, but we're already seeing some major advantages where compared to E. coli, you can make about 4 times more protein per culture, and as I already mentioned, it has this capacity to not only make the proteins, but to then secrete them out directly into the growth medium. So thinking about what a process looks like where you have to harvest cells, crack open the cells, and purify the protein away from all the other proteins in that cell. This just dramatically simplifies the downstream processes. Just in closing, I just wanted to point out, we're also thinking about genome design principles for eukaryotes like yeast, and to go along with the Vmax theme, we also have Kmax. So Kmax is based on Clivaromyces marxianus, which is the fastest growing eukaryote. It's a thermotolerant yeast with a fairly small genome size of only 10.9 million base pairs, but unlike the bacterial systems, it's spread across 8 different chromosomes. So thinking about our design, build, test cycle here, so with design, there's just an enormous amount of information out there in the literature where we can leverage comparative genomics in Saccharomyces cerevisiae and Schizosaccharomyces pombe, where a lot of genes that are non-essential are already known. And then also doing this random mutagenesis again, understanding what genes can be knocked out. But then, we can build chromosomes. We've shown all the chromosomes in this Kmax organism are within reach of synthesis for us. We very quickly, in just a matter of weeks, synthesize this one chromosome, chromosome 7. And then with test, so unlike with the bacterial system where you're completely replacing a genome all at once, this would be more of a chromosome transplantation where you swap your minimized or modularized or minimized and modularized yeast chromosome into Kmax and do a swap of that chromosome and then see if the cells are viable. So this is an exciting program that's just getting underway at SGI at the moment and JCVI. So in closing, I'd like to, there's so many people to acknowledge. This is a project that's been going on for quite some time at JCVI, Synthetic Genomics, and at SGI DNA. You see the names here. I could tell you something amazing about each of these individuals, but let me just once again thank and acknowledge our genomics and synthetic genomics pioneers, Craig Venter, Ham Smith, and Clyde Hutchison. So with that, thank you very much for your attention.
Thank you very much. We will then have a short break. We'll start again at 4 o'clock. At 4 o'clock there will then be an opportunity to ask questions to all the speakers who will be lined up here on stage. And so there will be a panel. And so you can either drop questions in the jar here, but you can also, of course, address them directly to the speakers. We will focus, when we have the Q&A session, we'll focus on taking questions from the young audience.